battery power left.
Yeah, now it's
Yeah, can you see my slides, Subrata? Yeah. Yeah. Okay, thank you. Good evening, everyone. Thank you, Subrata, for the kind and Professor Pradeep for the kind invitation. It's always a pleasure to talk in front of Professor Pradeep because there is always something new to learn and always there is a new perception. Always you get it when you present your work in front of Professor Pradeep. So thank you very much. Just take 1 second because I still like to recollect my first interaction with Pradeep, Professor Pradeep. It was in 2002 when I presented my PhD result as a poster in CRSI symposium in NCL Pune. I remember that long interactions with him that time, and that was really kind of new experience for me. And so it's always nice to learn. You are someone who we all look up to as a role model. Thank you for the kind invitation, probably. Yeah. So let me quickly. I hope everything is clear. Just let me hide this. What we have been, what I will share with you today is some of the research we have been doing on one kind of materials, which is called supramolecular polymers, which we really get. If you consider this as one of class of materials, if you really like emerge over the last 2 decades as one of the most promising candidate for, if you look into an adaptive or self-healing material. When we design this material, we take a lot of inspiration from biology. So I will let you know how do we take this inspirations from biology and how we adapt in synthetic systems. And ultimate aim is, can we have lifelike properties in this kind of synthetic material, in this kind of soft materials? So this is some of the directions we are pushing in. So I'll share with you some which direction we are going. This is a field which is complex. So we are learning along with the growth of this field. We are slowly learning and we are trying to push this field into more and more complexity and more precision. So that's something which I would like to share with you today. So before we start, let me tell you our group is working at the interfaces of organic synthesis and molecular self-assembly with the ultimate aim of functional soft materials. And one of the components always is a biconjugated system so that we can get directionality functions in terms of its optical and electronic functions. And also a biconjugated system, one of the advantages, we can use it as a nice probe to monitor the self-assembly. So with that context, most of the building blocks or monomic building blocks, if you see my systems are, our systems are kind of biconjugated systems. So one active area of research is organic photophysics. I mean, especially the photophysics from organic molecules. So we are one active area we are recently entered into is making use of ambient organic phosphors, organic triplets, and explore properties like thermally activated delay fluorescence, CPL, and so on. This is one kind of active field, but I'm not going to talk about this. And we also, since we work on supramolecular chemistry, we know that they are highly dynamic, they are adaptive. So we make use of such reversible properties of these materials, supramolecular interactions to make very smart materials like sensors. Advantages that they have will have very fast response. But as I mentioned, I'll be talking about the supramolecular polymers, which we have been working over like last 1 decade, where we are trying to get control on its structure and temporal aspects of these supramolecular polymers. So let me introduce this class of materials, especially for the sake of students. So as you can see in this animation, if you put a molecule which can form directional non-covalent interactions, in this case is a hydrogen bonding interactions for this molecule. If you put it into a solvent, they can come together by very weak interactions and they can make large structures. And if you look into the structures, they are pretty similar to the classical polymers in terms of its large structures. In other words, they are micromolecules. But in terms of dynamics, if you look into it, they are pretty simple to protein assemblies. So this is a video animation, is another animation of microtubules where tubulin protein monomers are coming and disassembling, which is serving many functions in our cellular systems. The only difference between the classical polymers and in this kind of non-covalent supramolecular polymers is they are highly dynamic. So there is a monomer exchange dynamics between the backbone of this polymer and the surroundings. So that will give you a lot of properties like self-healing, adaptivity, self-repairing, So these are the one of the advantages of the supramolecular polymer. So last 2 decades, a lot of research has happened in this particular field, and I just want to tell you a couple of very unique features of this, which came from different groups in the world. One of the earlier report was making use of these dynamics to make self-healing plastics. There is already a startup in Europe from Suprapolix. So this is a quadrupole hydrogen bonding motif based. There are 4 hydrogen bonds based supramolecular polymer, and as you can see that if you cut into it, the dynamics between the monomers bring them closer and make them self-heal. And this is a slightly different mechanism when you could look into the classical self-healing polymers, where you need a catalyst, and when the crack initiate the catalyst, the self-healing happens. But here is just like biological systems, is purely kind of dynamics between the monomers. They are bringing this self-healing happens. And very recently, I'm like, so a couple of months back, a few months back, towards the end of the last year, there is also now a lot of interest towards sustainability and making use of the supramolecular polymers towards recyclable plastics. We know that the interactions are weak in between these monomers, so we can always think about recycling them and upcycling them and then use it for various purposes. This is something many groups are working towards. Can we make use of these supramolecular polymers as something kind of recyclable plastics? So as you can see that the material offers a lot of opportunities, but the question is, can we really replace the classical polymers with this kind of supramolecular polymers, which is always a challenge. What we have been trying to do, along with many other groups, there are some world leaders in this field, like Bertmeyer, Takuzo, Ada, Fruchtner, Wuttner, and Agai. So what we have been trying to do is, if you want to really get a complete control on this class of polymers, it is very important to understand their mechanisms, to control their structure, and we have to go to multi-component system, just like classical polymers. We should get temporal control on these systems. So what we have been trying to do is trying to do this supramolecular polymerization process. So 10 years back, we have been looking into the mechanistic aspects. So we spent a lot of time in understanding how this growth process happens. And then very recently, we have been trying to look into control its molecular structure in terms of its dispersity and in terms of its molecular weight, because that is an important aspect. That precision is an important aspect if you talk about functions out of this kind of materials. And now we are trying to expand the structural diversity of this kind of polymers. We are going to multi-component systems. We are trying to get topology control on this kind of systems. And more importantly, we also look into temporal aspects of this kind of supramolecular polymers. So throughout this talk, I will try to give a kind of thought process, how we are pushing this field forward, and then trying to impart these kind of lifelike properties into this kind of systems. So precision is the key. The first we thought it's important that we should get a complete hold on the synthesis of these molecules, synthesis of these kind of polymers, so that the precision is the key in this kind of system. So how do we do it? So the thought process was, which was the game changer, I would say, because earlier when we tried to, many other groups tried to make these kind of systems, we were trying to make them under thermodynamic control, like any other materials. But once we make them under thermodynamic control, one of the issues we faced is that the product is always highly polydispersed, because you don't have a real control on the growth process. But if you look into the biological system, as I mentioned before, most of these biological self-assembly process, like protein self-assembly and everything, is under kinetic conditions. And we know that precision is ubiquitous in nature. One of the ways we have been trying to do is to switch from this thermodynamic control to kinetically controlled synthesis, and that is exactly similar in classical polymers also. When if you look into atom transfer radical polymerization, ATRP, when classically covalent polymers are synthesized through kinetically controlled ways, then we could achieve the precision in the synthesis of these structures. So this is one of the thought process, switching from thermodynamic control to kinetic control. Just like I mentioned at the beginning of my talk, it is all about bio-inspired designs. And this is one of the key aspects, that we adapt the bio-inspired kinetically controlled synthesis for the precision control in this kind of supramolecular polymers. Let me explain how so this slide is important for the subsequent understanding. So how we achieved in a kind of cutting the long story short, so we call it as a living supramolecular polymerization. Living in the sense is nothing to do with the biological structures. What it means is the chain ends or the chain backbone of the supramolecular polymers are always living. That means they are always active and they are not dead. So once they are not dead, what happens is we can always keep adding the monomers to the backbone. So how do we achieve such a living nature into this kind of supramolecular polymer? So what we have done is we introduce a kinetic control in this growth process. So there are multiple mechanisms known in the growth of supramolecular polymers, but let us stick into the typical well-known nucleation growth mechanism, which is similar to the chain polymerization in classical polymers. So we know that there is a nucleation step, which is energetically unfavorable, and then there is a propagation, and we call it as a elongation or growth of the polymers. We know that step is energetically unfavorable, but even if you look into most of the synthetic system, the energy required was very small. So as a result, most of the supramolecular polymerization, when we try to do, they were really spontaneous. The growth process was very spontaneous. So we could not get a control on the growth process. So one of the key thing was to delay this nucleation step. So how to delay this nucleation step? So we approach many kinetically controlled manner. So if you look into biological systems, so amyloids growth also, amyloid fibril protein assemblies growth, there are metastable phases in between, like microscopic droplets and so on. This provide a kind of kinetic barrier to the growth of this supramolecular, this kind of protein assemblies. So we try to mimic this kind of bio-inspired design into synthetic systems. So what we did, it took the monomers, we purposefully introduced this kind of nucleation pathways or metastable states in this kind of synthetic systems. So the whole idea is that you provide a kinetic barrier to the growth process. So with this, we can kinetically delay the growth process. Then once we have this control, so always what happens is once the polymer is formed, if you introduce additional batch of the monomer, we know that independent nucleation is slowed down because there is a kinetic delay, kinetic barrier involved in that. What happens is it always look to grow or seed in the presence of an existing backbone. So what I mean is if you introduce a monomer, they are in a kind of dormant state. They cannot start its own nucleation, but they will prefer to grow on the existing chain. So that give you the living characteristic to your growth chain. I hope you got it. So what we purposefully do is create a dormant phase for the monomers and introduce into the systems so the monomers keep growing not by its own, but it grows only at the ends of the chain. So that was the thought process, and that way we can get living polymers. And once you get the living polymers, it's very easy to control the growth, the molecular weight, because simply the monomer to seed ratio will give you how much monomer you introduce and how much seed is present. That will, you can predict roughly the molecular weight of the polymer. So this is, we call it as a living supramolecular polymerization. Just one example I would like to show you. This is the kind of systems we work on. So these are biconjugated N-type naphthalene diamides, which is substituted with both sides some kind of hydrogen bonding motifs. So what happens is if you put it into a solvent, this exists in a kind of folded conformation, which we call it as a dormant phase. And then that with the kinetic barrier, with the time, it opens up and grow into a kind of polymeric chain. So if you look into the growth process, which we can probe with any of the spectroscopic properties, because the intermolecular interactions are very sensitive. The spectroscopic properties are very sensitive to intermolecular interactions. So we can either probe circular dichroism, absorption, or fluorescence, any of those things we can probe as a function of time. Then we get beautiful sigmoidal curves like this, which is very similar to the protein self-assembly. And then we can use the models which are there in protein self-assembly and to fit into the growth process and get all the parameters out of it. And as you can see that once this kinetically controlled growth process sets in, the polymers we get it quite narrow disperse. And we can repeat this seeding process multiple times. And since it is a living in character, it will grow only at the end of the polymeric chains, and you can keep on increasing the length of the polymers. So this was very nice. So this gives you a kind of precision control on the growth process, which was not there when we tried to synthesize these molecules under the thermodynamic conditions. This is to introduce how we achieve the growth process. So moving forward, so this is, as I mentioned, is very easy now to control, very easy. To a certain extent, we can control the growth process and get control molecular weight and disposity. So we can always think about now we have controlled growth, why we can jump into basically multi-component systems. So far, we have been working on one component systems. So basically now there's a possibility that we can send it to, we can think about more than one component. So we have been looking into naphthalene diamide systems because naphthalene diamides, the beautiful work from Mattile and Frank Wuttner, one of the beauty is that just by minimal changes in the core, we can change the HOMO-LOMO gap. And so we can get diverse optical and electronic properties if we work on these kind of systems. So we can think about more than one component, different molecule, and we can make blocky systems like this, because once the growth process is complete with one component, you can always come back with come with another component and it starts growing. So this gives you a kind of blocky structures, but from an organic materials perspective, if you look at it, they are very similar to organic heterostructures. Similar to the inorganic heterostructures known in.
So one quick question I wanted to clarify.
Yeah, sure.
So when the dormant state, how do you turn it on? What does that trigger?
So this is, so under, there are, you don't need an initiator there. What happens is you just delay a kinetic barrier there. So it's a metastable phase, and with time it converts into it. So how deep is the kinetic well? It depends how long is your lag phase. Okay, so there are structure property relation studies. It depends on the length of the chains, string size. In this case, in this particular case, it's an intramolecular hydrogen bonded system, which is the dormant phase, which depends on the linker strength, linker length, and so on. So it is not a deep kinetic trap. It just provides a kinetic delay, and once it initiates, then the propagation happens quickly.
Yeah, so there is a molecular change that is happening, right?
Yeah, there is a conformational change in one case. We can also think about just like amyloids off nucleation pathways. So there are a couple of different strategies we do it.
Time is enough to switch the conformation.
Yeah, exactly. And once it happens to a person, then there is a kind of cooperative effects in then switch the equilibrium to the other side.
Thank you.
Yeah. So this gives you an opportunity to get organic heterostructures because in the past there was no way to make such structures because we do not have control on the growth process. And now, so this gives an opportunity to make organic heterostructures like this, and we are collaborating with physics groups to look into the exciton migrations and all other things from this kind of single organic heterostructures. So what I started is a controlled growth. I mentioned that we can also go into one-dimensional multi-component systems to make blocky structures, just like classical polymers and organic heterostructures. So what very recently, one more, I just want to show you one of the recent result in a little bit more in detail before I go to the temporal control. I will take one example in detail from a structural control point of view and one from a temporal control point of view. So if you want to really replace these supramolecular polymers as at least put a competition to classical covalent polymers, is very important to expand the structural diversity of the supramolecular polymers, because so far we have been talking only in terms of primary nucleations and one-dimensional growth. But we know that in classical polymers, there are branch systems, there are star polymers, and so on. So how do you expand the dimensionality, or how do you expand the structural diversity of these polymers? So as I mentioned in the beginning, always we look into bio-inspired designs. And if you look into biology and biological system, especially protein cells for self-assembly of amyloids, the autoproliferation of this happens not by a kind of primary nucleation, but something called surface catalyzed secondary nucleation. So what happens is primary parent fibrils are formed, and the surface is acting as, which is well known in crystals as well, where surface is acting as a kind of catalyst to make more and more daughter fibrils. And we thought probably we have to move away from primary nucleation to create the secondary nucleation sites if you want to make branch structures or orthogonal heterostructures like this. So this is another thing we started during the COVID time, a lot of reading process that time, and then we got some clue, and immediately after the COVID, we started working on this kind of secondary nucleation. And thanks to, again, protein self-assembly beautiful models, if you plot the log plots of T lag and versus the concentrations, the power law, the scaling exponential tells you exactly whether it follows a primary nucleation or secondary nucleation. And if it is less than 1, generally it is primary nucleation, and it is greater than 1, it is secondary nucleation. So we have a lot of kinetic analysis have to be done. There comes the importance of our spectroscopic probing, because to differentiate between the primary and secondary, it's important to have enough data points at all stages of your growth process. And the spectroscopic probing really helps to get that differentiation. So what we have done is, we have been looking into the secondary nucleation, and we have got control on structures. We are able to now growth structures like branched, kind of orthogonal, and so on. I'll just take one example. How do we get control to explain you in detail? How do we get control over primary and secondary nucleation? Because again, precision is the key. If you want to really, so it is very important that you design your monomers in such a way that you can selectively bias the growth process either to the primary or secondary nucleation. So how do we tell these molecules to follow selectively to one of these pathways? So that was the key. We use a couple of different ways. One is obviously we like its chirality, and the other one is we make use of different molecular organization. So I'll take one example with chirality. So Savikan has come up with very simple naphthalene diamide systems, which has having a peptide side chain, which is well known to form beta sheet kind of structures. So this forms also a nucleation growth self-assembly, kinetically controlled, if you look into it. So what happens is, since there are two amide chains and chirality present, if you take both enantiomers, there is a chirality mismatch. What I mean is, so if you take nuclei of homochiral nuclei of one of the component, and if you add the similar monomers, similar chirality, homochiral seeding we call, if you do that, there is no chirality mismatch. It prefers a primary nucleation. And as soon as we take the opposite enantiomer, which is having opposite chirality, and then try to do a heteroseeding, what we see that there is a chirality mismatch sets in. So the primary nucleation pathways are completely blocked. Then obviously, since the molecules are in a kind of dormant phase, it looks a way to bypass the nucleation barrier, and obviously it looks to grow on the surface of the primary fibrils. And that worked exactly, I mean, very well. And if you do detailed studies, we can nicely fit the kinetic profile, and homochiral seeding nicely fit into the primary nucleation, and heterochiral seeding nicely fit into the secondary nucleation. And seeing is believing. So when we look into the fibrils through AFM or some other techniques, obviously one has to look into image them in solution. That is one of the challenges. But AFM, we don't do that. It is a dry state. There comes the importance of cryo-EM and other techniques. But then we do see the secondary fibrils growing on the first fibril. And the ratio of the secondary fibril depends on the enantiomeric excess, as we expect. But then we wanted to really visualize them in a better way in solution, because these molecules are in non-polar solvents. We tried to mimic them in polar solvents as well. So cryo-EM right now, we are not able to use in the systems, but we wanted to really prove it in a slightly better way. And also, so this Saikat has tried to now differentially substitute this core of this naphthalene. This is an ongoing project. So naphthalene diamides, with one is with oxygen and one is with sulfur. And oxygen will have a green emission, and sulfur will have a kind of reddish emission. The way it is organized. So we try to do exactly similar chirality controlled stereoselective pathways. So if you take homochiral, it works exactly same. You can see the beautiful kinetic profiles for the heterochiral comes in. And if you look into the scaling exponential, it is minus 2.9, which clearly suggests that it is a secondary nucleation. So here again, if you take the homochiral, it goes to a primary nucleation. And if it is heterochiral, it goes to a kind of secondary nucleation. And this is super resolution images. I hope you would be able to see that there are two fibers of different colors are sticking to each other. So this is a beautiful demonstration that indeed we have a perfect control on the primary and secondary nucleation pathways. And then the beauty is that we can now selectively elongate one of the component with respect to the other, because we can do stereoselective seeding, subsequent sequential seeding, and we can selectively pull, elongate one of the fibers with respect to. So this is very interesting. So this is clearly demonstrate that we have a good control on the primary and secondary nucleation pathways, and we are now able to make something is in organic photovoltaics well known is double cable polymers, because there is a donor at the backbone and acceptors as kind of pendant. So this create kind of orthogonal heterojunctions, which is good for charge suppression and so on. So what we have done now is we have made a kind of supramolecular analog of that by clearly controlling the primary and secondary nucleation pathways. And again, these now nanostructures are with Arindam. So he's now trying to look into the energy transfer processes. The beauty is that we can get different topology by varying the different ratios of donor and acceptor. We can either make this kind of topology where acceptor is only at the end, or we can also make this kind of topology it is at both places and so on. So this works very well, the control on the primary and secondary nucleation pathways. So we are quite excited about it, and we are trying to extend it to now PN junctions, because these are energy donor and acceptors. So we can also modify the monomeric structures into kind of electron acceptor and electron donor. And so we are now currently synthesizing those molecules to demonstrate these PN junctions. So quickly to a couple of more different directions we do before I show one last example on temporal aspect. So one more thing is, we know that liquid phase separation is, as I mentioned, we are trying to mimic most of the lifelike properties in this kind of supramolecular polymers. And one of the recent sensation is mimicking liquid phase separation, which is membrane-less organelles in synthetic system, which people call it as conservates or droplets and so on. So we know that liquid phase separation is kind of membrane-less organelles are there. They are highly dynamic. They form from the liquid phase separation because of the large proteins and intrinsically disordered proteins and so on. So apart from a kind of, I mean, if you look into recently, how people are trying to make them in synthetic system is using big macromolecules, especially charged molecules. There are three ways, like simple conservation or complex conservation or auto complex conservations. So they are making use of large charged polymers, and then in presence of certain solvents, they try to microphase separate and then make this kind of dynamic fluid phases. And people use it as a nice way in synthetic biology as like a kind of, it is quite different from vesicles. It doesn't have a membrane. So people are using it for studying as a reaction container or trying to look into the fusion of this kind of droplets and so on. But our perspective was to see whether we can also make it from small molecules, larger than charged molecules, because supramolecular polymers can play an important role. So the Satyajit was trying to look into this kind of systems very recently. And we have a couple of very interesting designs that I'd like to share with you, because this is another kind of emerging field. We can see that lifelike properties can be also looked into from this kind of systems. So this is a slightly different molecule. You can see that we try to incorporate that charged auto-complexation design of classical polymers into it. There is a cationic group and an anionic group. And then we see that immediately it phase separate in water to form these kind of droplets. And they are highly dynamic droplets. We can confirm that they are indeed conservate droplets from TM and so on. And then we do fluorescence FRAP studies, fluorescence recovery after photo bleaching, to ensure that they are not solid structures, they are indeed dynamic phases. And but then obviously, since there are pi conjugated systems, with the time the crystallizations, pi taking sets in and crystallization starts, they form into 1D fibers. So it was very nice to see that we could achieve this kind of liquid droplets from liquid phase separated droplets from these kind of pi conjugated molecules. We have been working on it. I don't have time to go into the details. This is another recent study from our group. So there are certain design principles if you look into the peptides which forms this kind of droplets, which is they call it as sticker spacer sticker model. So we try to mimic that model in small molecular systems. And then in this cases also, as you can see that the droplets are formed, and we try to couple into a kind of reaction. In this case, is a boronic ester reaction, so that we can transiently form this kind of droplets. So it forms, dissolves, like kind of things. This just to show that this is another field we are getting into it to make sure that we can also make these kind of systems with these small molecules. Then just quickly, since time is running out, I will just skip the next part and then go into the last part of my talk, which is so far I've been talking about structural aspect. Now, another aspect we have been looking into is temporal aspects, or making these molecules away from equilibrium and so on. So if you look into, so far I have talked about it, we have nice molecular designs for controlling the structure of these molecules, structure of the supramolecular polymers. But one of the aspect is they are not, predictive design is one of the challenge. What I mean is that one design nicely work for one class of molecules, but when you go into another class of molecules, the energy landscape is so diverse. We have to standardize it again and again. So it's very important that come up with the common strategies to make these systems highly precision. Synthesis is kind of common strategy. So again, if you look into nature, they work in a slightly different way. If you look, this is another nice animation of actin self-assembly. They are coupled to reactions. So chemical reaction coupled. For example, ATP synthesis connected to feedback loops. ATP synthesis controls the growth of ATP self-assembly, and once ATP get hydrolyzed, it disassembles. So one of the characteristic thing is it has a reaction coupled nucleation growth if you look into most of the protein self-assembly. And also there are feedback mechanisms between various auxiliary proteins because it is connected to reaction networks. So they are living in character and so on. So we have been looking into these kind of systems. Can we mimic in synthetic systems? And once we can do such systems, probably this is a kind of generalized strategy to make precision supramolecular polymers. And for a chemist, it's not only enzyme. We know that chemist has a lot of opportunities like chemical reactions, redox reactions, and so on. So the opportunities are many. So that's why we have been trying to work on this kind of reaction coupled fuel driven cooperative growth. Fuel, I will take it away because there are a lot of debates on the use of fuel, but reaction controlled cooperative growth is something which we would like to implement in this kind of systems. So the whole idea is that we have inactive monomers, then it coupled to a chemical reaction, it converts to an active state. So this is a dormant phase, and that is conducive to the assembly process, then nucleation growth assembly sets in. The beauty is that we can always couple it to another reaction network, which is a deactivating reactions, and then we can probably push it into a kind of non-equilibrium regimes and so on. So this is something which we have been working on. I will show you two examples how we are trying to do this. And that second example, we are quite excited about it, and we are going on. We are doing some aspects of it. So this is one of the recent JAX paper we published, where Anshuman and Saikat could couple this growth of these supramolecular polymers into an enzymatic reaction network. So this is a molecule here. This is an NDI molecule, naphthalene diamide molecules, which has both sides ganidinium receptors, very similar to the arginine receptors. And the beauty is that it is selective to ATP binding to form this kind of 1D self-assemblies. So what we thought is we can always use an enzyme to in situ generate ATP from ADP in presence of some of the primary substrates like phosphocreatine. And then once we do that, the rate of the enzymatic reactions will be the rate of your growth. Because as soon as ATP generates in the system, it will bind to a monomer and the growth process sets in. So if you look into here, if you simply add ATP into the system, the growth process is spontaneous. Now what we have done is we have coupled this into an enzymatic reaction network, and you see that as the ATP keep generating, there is a nucleation growth sets in, and the growth process become kind of controlled growth process. And again, a lag phase and a growth phase sets in. So the beauty is that we can control the lag phase, we can control the growth process, extent of growth by changing any of this component, either the enzyme concentrations of the primary substrate concentrations or ADP concentrations, anything. And one more thing is we have done a lot of enzymatic assays, like luciferase assays and everything to find it out whether the reactions are coupled to each other. What my point is that coupling is when we say reaction coupled, it's very important that there is a reciprocal relations between the enzymatic reaction and growth process. What I mean is, as the supramolecular polymers start forming, it should influence the enzymatic reaction rates and vice versa. Okay, so what I mean is they are mutually coupled. The reaction and the growth process should be mutually coupled. So what we found that there's a positive feedback mechanism exists as we are forming more and more polymers. The enzymatic reactions becomes faster because we found that some of the enzymes are binding to the the polymer backbone. So what I would like to say that there is a positive feedback mechanism exists here. They are really reaction coupled process, just like in actin self-assembly. So as I mentioned, so now once we have it, we can change any of the component, like we can change the primary substrate concentrations or enzyme concentrations or any of these concentrations. We can predict to a certain extent growth process. So this is quite different from using an energy landscape or a metastable phase, where as I was answering to Professor Pradeep before, it's not easy to predict how much lag phase you would like to have. It depends on the energy landscape, which is very sensitive. And here, probably to a certain extent, we can predict because it can be controlled by one of the chemical component. So that's something nice. So the last 5 minutes I would like to show, which is an example where we are trying to push this reaction coupled system to the next level. We know that microtubules is one of the beautiful example of a reaction coupled protein assembly. But one of the characteristic of a microtubule is an oscillating system. Assembly, disassembly happens under autonomous conditions. So there is a tubulin monomer, which is an inactive active monomers. It forms microtubules, and then once GTP is getting hydrolyzed, the strain builds in the microtubules, and there's a catastrophic disassembly happens. And these oscillations keep on happening. So the point is, can we create a kind of artificial synthetic assembly which is autonomously oscillating like this? So this was a dream for us for some time. And then so we have all the ingredients, like we could couple the reaction into the growth process. We can probe the mechanism. So how do we achieve such a autonomously oscillating assembly? And if you look into microtubule, so there are important characteristic if you want to get this kind of oscillation, autonomous oscillation. It's not like you provide at the end of each oscillations additional energy or anything. It has to be continuously assembly, disassembly process should happen. And that's one of the characteristics thing in. There are multiple requirements for this. Okay. One of the important thing is it has to be reaction coupled. We have done this. And important aspect is it should have kinetic asymmetry. What I mean is the assembly pathway and disassembly pathway should be different, because otherwise even after one cycle it will equilibrate. And there should be selective reactivity for the monomeric phase and assembled phase. These are the couple of important criteria for kinetic asymmetry. That means assembly in an assembled state, monomer reactivity should be different than the monomer. That is very important to impart this kinetic asymmetry into the systems. And once we once these conditions are satisfied, one should be able to get an oscillating assembly. I know that I'm going a bit fast on this, but believe me, so we are trying to, Vandana has been trying for last couple of years, 2 to 3 years, to make such a system. And she has come up with a beautiful concept to get, because we can couple the reactions, that's not an issue, but what is very important to get a kinetic asymmetry in the system. So what she has come up with a very nice system. So this is again one of the naphthalene diamide appended with peptide systems. So this forms a nucleation growth mechanisms. And so there is a positive feedback mechanism here, because it's a nucleation growth mechanism that gives a positive feedback. What is important to connect is a negative feedback. So how do we do it? She has made use of photoredox assembly for a reason, because what happens is, if she do this, continuously shine light into the system in presence of a sacrificial agent, in this case it's a DMF, what happens is the energy levels are in such a way that this photoreduction brings these systems only to an NDI radical anion, only an one electron transfer process. It never, if you do a chemical reduction, it immediately go to a kind of dianion. Dianion means there is electrostatic repulsion, assembly will immediately disassemble. So what she has done is she has used a DMF as a sacrificial agent and then did photoreduction of these assemblies. What happens is it only goes to a radical anion, and radical anions still stacks because there is interactions between them. So they keep it in the assembled state. But what happens is in the assembled state, when radical anions are stacked like that, there is a disproportionation reaction sets in, which leads to a kind of NDI2 minus in the systems. And this disproportionation happens only in the stack, because in solution it can never happen because it's a photoredox. The energy levels are in such a conditions that monomer will never go to 2 minus. So monomer 2 minus happens only in this stacks. That is what is the key thing in this case as a kind of kinetic asymmetry, what I'm talking about it. So it has a selective reactivity for the monomers in the solution and in the assembled state. So we ideally, basically so once that disproportion happens in the stacks, it goes to a kind of 2 minus, and then this process continues because there is continuously in the presence of oxygen and light. So this assembly disassembly process keep on happening, and then we are able to get these kind of oscillations. And as you can see that this is quite dampened oscillation. There are multiple reasons for that. I will come back to that in 2 minutes. I have to wind up in to Subrata. I'll take 2-3 minutes to wind up this. So what we could do is we could really study each and every step in this particular case. We are able to now study the positive feedback mechanism. There is a kinetic asymmetry, and there is a delayed negative feedback in the sense there is a radical anion goes to dianion, which leads to disassembly. So there is a nice delay feedback, which are all characteristic similar to the microtubule assemblies. So we can look into each studies, each step selectively and so on. So as I mentioned, kinetic asymmetry is the key, and we are able to get all those things kinetic asymmetry because of the selective reactions in the assembly state, which is the disproportionation reactions. And obviously, we are very disappointed when we have a dampened oscillations. And so we have approached, because the point is that we get sustained oscillations only at a small region, which is highly sensitive to all these redox states and oxidation states. There are multiple steps involved. So we would like to find out exactly that regime where you get sustained oscillations. So we needed help from the mathematicians. So they have done detailed kinetic modeling. They are expert in looking into out of equilibrium systems and reaction coupled patterns and so on. So they told us where to look exactly. And now we are coming back and looking at to the regions where ideal concentrations and ideal light intensity one has to look at it. And as you can see that the oscillations are indeed sustained to some extent. There's something which we are quite excited about it, because this is possible only because of the detailed understanding on the mechanisms, and we are able to couple a positive feedback mechanism and negative feedback mechanisms. So if slight variations you go here, you get dampened oscillations, you get steady state, you can get sustained oscillations if you are in this circle. So we are still continuing. So we believe that is a kind of, at least structurally, is a kind of oscillating microtubules. Why we call it as, because periodic doubling, chaos, we move around little bit here and there, we get it all the kind of patterns which ideally microtubules have shown in literature. So we are quite excited about it. Now, most important thing is obviously, can we pick up this spatial temporal dynamics in visualization as well, which is a big challenge. We are not able to do it so far. So we are now tagging with systems like sign-in kind of dice, so that we are able to pick up this dynamic instability. Like this is a beautiful video for the microtubules people have obtained, that fibers are growing and autonomously is going. Our dream is to visualize something similar in synthetic system as well. So we have been working towards it. I think it's time to conclude. So what we have been trying to do is get a better control on the precision in structure and dynamics of these systems, trying to get more and more lifelike properties, not to make a life, but to get smart functions out of these kind of materials and to get a better control. So we look into the spatial temporal patterns, and very recently we are now trying to look into can we integrate these multiple assemblies into a kind of cell-like environment. So we are using now protocells and putting these assemblies inside these protocells. And then we realize that the pathways are changing, because once it is in a constrained media, we know that that's why I skip those slides. There are other issues like micromolecular crowding. What is an effect of a macromolecules on the growth process of other systems? So we have been starting looking into those aspect as well. The effect of crowding, when there is a space is restricted to do this self-assembly process, how it is affected and so on. So we are looking into more and more different directions in this field. So it's a slow learning, as I mentioned, it was a slow learning process. We have started with structure, length and dispersity, very simple problems. We now extended it to multi-component systems. Now we are trying to improve the hierarchical complexity to branch structures and other topologies. We are now very recently more and more interested, can we really get the control on the temporal aspects of also these kind of supramolecular polymers? So when we started our research 10 years back, 15 years back, this was one of the articles appeared in Science, which really says that self-assembly is one of the what we don't know issues. And this article really pushed for getting a better precision. And this article clearly tells us, look at biological systems if you want to get inspiration. And that's what exactly we have been trying to do. So we were trying to look into the precision, structural diversity, and some aspect we have to improve is obviously the predictive design in this kind of systems. These are the people who contributed this supramolecular polymers field. They are wonderful students, and right now it is spearheaded by Saikat and Vandana, and most of them have really contributed to this growth, understanding of this supramolecular polymers. This is the current group, and a lot of collaborators, especially molecular dynamic simulations. We have a lot of collaborators like Bala, Juni Pawan, Jagannath Mandal, because one of the complexity requires certain understanding of many processes, and we use them. We use the help of molecular dynamic simulations, and we have collaborated from physics lab, and obviously super-resolution microscopy Sarit is helping us, and we also have some collaborations from abroad. Thank you for your attention, and sorry I took 10 minutes extra, I guess. I would be happy to answer any questions. Thank you.
Fantastic, as usual. As I missed a few minutes of your talk because I was just about to get into the hotel and connection was lost in between. Now, a couple of things that come to my mind is, first, if you are talking about biology or connections with biology, any chemical system moment, say one goes into biology is scalable. You can have one person or a billion people. Organization can grow. Now, what happens in these systems? Can one scale it up?
Yeah, it's a good question. It's indeed possible to scale it up. I don't see a problem right now. But what was important for this field so far is how to do this. Okay, so we were concentrating on that particular aspect rather than really go to the next scale. So that's something which we have. But is according to me, it's indeed possible to scale it up.
No, why I asked this question is that any oscillatory system, towards the end you were talking about oscillatory systems. If you take that as an example, I am not aware of chemical oscillations happening at mega scale. They do happen in bottles.
Yes.
They may not go to very high scales. Of course, there are chaotic behavior. You can model climate, etc., of this kind. But are there chemical examples? So this is one thought that occurred to me as today's world, of course, everything is an industrial relevance that comes in. So therefore, the question of scalability comes. Are there your nucleation growth control phases? Are they all if you give it to a chemical engineer, can they process it as a unit operation and expand it? So
Yeah.
that,
I mean, like, obviously there will be challenges. Completely agree to that. In the last slide, I have shown one example. So I'm like molecular oscillations at a reaction levels were well known. I'm like, what we could show is couple it to an assembly. That is obviously the next hierarchical level one talk about it. Just like the microtubule. So the last slide, I mean, I show about spatial temporal patterns, something which is on a slightly larger scale. I'm not in the, definitely from a kind of chemical engineer kind of scale, but you could see that the spatial temporal patterns is originating from these oscillating assemblies. So that is, we could show it in a slightly, not in a kind of cuvette. It's a kind of petri dish kind of scale. I mean, still the oscillations are happening there. But obviously, yeah, I'm like the processability level, next level, we have not thought about it. I'm sure there will be challenges to take it up. But then, yeah.
I will take two more questions, the short ones, and then leave
Yeah.
it to Breto. So you assemble, disassemble, you create oscillatory patterns. Obviously, you will be releasing energy.
Yeah.
Absorb, release, and all that. So this would be like biological systems too. Now, are you monitoring this as well? Are you getting for a unit step, the energy involved? Are you in a position to monitor?
Yeah, I mean, like, obviously one has to monitor, but we are not in a position to assess that, how much quantification we can do, because one has to look into microcalorimetry or one kind of experiments involved. It's indeed possible to do it one of the challenges to do it in the solution phase, a diluted phase. But obviously, I completely agree to you that quantification is very important, but we have not done so far because we were looking so far. I'm like, this is a kind of, this itself was challenging to get autonomous oscillations in a kind of open systems like this. People could achieve this is under microfluidic or in a kind of external forces. What we have done is really coupled a reaction to it. And one key thing was the kinetic asymmetry, and then we are very happy that we could find a mechanism to do it. But then we need to quantify each step through techniques to energy. I mean, we have not done it. Yeah.
The last thing that I want to do, biology, of course, obviously means a lot of functions.
Yeah.
So one function is turn on, turn off, luminescence, on, or hydrophilicity, hydrophobicity. To what extent can you create functions as part of this?
Yeah. So I could not show one more slide. So what we are now trying to do is, obviously the catastrophic disassembly of microtubules generates a lot of forces. Okay. What we have now tried to see in this present case, how much force it generates. Okay. And then how we have done that's still going on. We are doing it in collaboration with a German group. So what we are trying to do is attaching a kind of fluorescent bead to it, where same way microtubule force generation has been calculated. So we are in the process, and we could see that the amount of piconewtons, whatever generated in microtubule, we are in the similar scale. But then that is obviously what functions comes out of it, we have to see. But then autonomous oscillations, I mean, like, apart from the spatial temporal patterns, right now I cannot think about a function. We were more curious about replicating this in synthetic systems. But one thing which we are looking into is kind of force generation in a kind of levels from these microtubules, at synthetic microtubules, I would say.
Thank you. Subrata, please take it forward. Yeah.
So anyone have any other questions? I have two questions actually, like regarding. Okay. Anyone have, or say like anyone have any questions?
Yeah, I want to go forward.
Yes.
Subrata, if you may allow me.
Yeah, you're on.
Okay, perfect. Okay, first of all, thanks, Professor George, for like, it's always a pleasure to listen to your talk, as there is always something top notch and like advanced science which will be coming. So I have few questions. I will start by the I want to extend that with what Professor Pradeep was asking. So
Sure.
we know about this molecular oscillatory systems. Like we have done some practicals in our like.
The B-cell reaction is well known. Yeah.
Exactly. So how different is the kinetics from what you are doing and what is the BZ reaction?
Yeah,
is?
so molecular oscillators are well known, as Professor Pradeep also mentioned. So what we have now tried to do is try to get into couple into a kind of assembly systems. But as soon as this assembly system, so far whatever attempts to make these oscillations, one of the key thing was that it equilibrates after one cycle, because it's not possible to couple most of the assembly disassembly pathways and everything is similar. So the challenge was one step further to bring that kinetic asymmetry and delayed feedback mechanism in the growth process. So that is something which we have achieved in this particular case. So now we are able to look into, I mean, apart from the molecular systems, we are now looking into a kind of materials level oscillations. So that is, we could do that way is quite different from that, from the classical BZ reactions or so whatever. Yeah.
Okay, I got it. For the second one, for making hierarchical architectures, right? Can we really combine this reaction coupled and covalent interaction coupled in the same spot and like do a very complex reaction to come up maybe with 4 polymers combined together? Is it really possible? And like,
I mean,
what is the current
it's
aspect on
indeed
that?
possible. I know that there are groups, people are trying to combine the classical, the covalent polymers along with the supramolecular approaches. But obviously one of the issue here is in presence of large molecules, large polymers. That is also another reason we are now looking into crowding effects and so on. So the enter dynamics changes, right? So because once you have macromolecules with you, so it's not easy to do this similar things with the big molecules. Obviously, the entire landscape changes. But indeed, there are people looking into it to get the best of the both worlds in terms of dynamics and classical polymer strength. Because if you ask me, supramolecular polymers, one of the challenge would be to use it for really strong, mechanically strong applications. So probably that is one way the combination of the both approaches may help. I don't look into that aspect yet. So,
I got it. Okay. The next one is about like how do you really map the free energy landscape experimentally? Like, can we really experimentally map that? Because you were talking about this like dormant state, then followed by that is going through some metastable state.
yeah. So experimentally mapping is obviously difficult to exactly calculate the metastable phases and all those things. But we do collaborate to get some idea. But that is a kind of rough idea. I mean, it's not easy because the energy landscape itself is complex. It really depend on the solvent temperature and so on. So the quantification is slightly difficult, but then we get a kind of relatively qualitative picture in that. Yeah.
I got it. And the last one is about
Sure,
okay.
go ahead.
The last one is about the Shoikov's work and Sobik's work, where like maybe I have missed the point. So when you are talking that for the surface nucleation, so is it like one by one polymers are sitting there, or is it like the other polymer is making a small nucleation and then that is sitting?
Yeah. So sorry that I could not explain properly. So what we are doing is that the primary polymers are formed, okay, and then nucleation happens on the surface of the existing polymer, okay, and then it grows along to the primary fibrils.
Okay, so the nucleation is happening on the surface, so that means like one after one is coming and they are getting joined.
Exactly. So it's a surface catalyzed nucleation. Yeah.
Perfect. Thank you.
We are blocking the primary nucleation pathways for that so that it exclusively go to the surface.
Yeah, that I have understood. But my question was like whether the nucleation is happening itself on the surface or like it's happening and then followed by it's coming and joining.
No, it's on the surface. It's on the
Perfect. Thanks for the information.
Okay, so my question is not mechanism, so I will take it to some other directions. Okay, so you are basically trying to make microtubules, which is like biologically active molecules in real life through chemical synthesis. Is it so?
Yeah. So as I mentioned, the functions is not a biological function. I'm sorry. So what I'm saying that right now what we have achieved is structurally and temporally mimic a microtubule it's special temporal dynamics. That's all we did it. At this point, I stop it there, but obviously the functions we look into it may not be a biological function.
So are they soluble? They are soluble in water, these polymers?
Yeah,
These
this
coil-
last systems are in water.
So how is the status of cryo? So we have
Yeah,
this.
so obviously the last one is a potential candidate for the cryo, because whatever the first molecules, secondary nucleation, whatever we did, it is a kind of non-polar solvents. But now more and more we are going into the water systems. But then cryo is obviously a potential way to look at these systems. But when it comes to the dynamics, again, like that's why we rely more on super-resolution microscopy and tag it with fluorescent probes and so on.
So people has done.
Well, we can do this thing as a time series in cryo also, right? I mean,
Yeah, I mean, I'm not aware. I mean, but yeah, that would be great if one can.
Cryo building. Yeah. So this is the cryo.
Yeah, I know. I'm definitely aware of it. That's
People have done a lot of work on these microtubules, actually. I know, cryo, single particles and all. But your systems are looking also promising because they are 3 micrometers. So you can see or you can probe
Yeah.
them on the TM. By the way, how is the stability of this your structures under the electron exposure? So
yeah. So good question. So it varies from the system to systems. At least the branched structures I showed where we made the structures, we could do the elemental map. I mean, like we could do the DAX mapping with the TL. So we could do that. So they were stable. So it depends on the systems.
okay, that's all. Anyone has any other questions?
Hello, sir. Soham here.
Yeah. So
So in one of your fast mode slides, you told that the molecular weight can be calculated from the number of seeding. So like, is it like this kind of reaction are 100% conversion? There is no side products or some
yeah.
percentage are
So
there?
very good questions. The two things one has to complete: completion of the product and the completion of the reaction and the yield of the reaction. So both are important. So most of these cases, we see that there is a saturation in the growth process where we assume that this is going to the 100% conversion. Okay, so that's what we assume. And then this obviously the size distribution calculation is not so straightforward as in covalent polymers. So here we have to do a kind of number average by counting the polymer, because we cannot use a kind of GPC or something, because it's a non-covalent interactions. Any dilution will dissociate your polymers. So we have to do the size distribution in a kind of batch mode. And what now we are getting is a kind of multi-angle size SLS, static light scattering, not the Mars detector, but where we can do a kind of batch reaction. So that's something which we are getting that will make our life much easier. Because we can calculate the size distribution in solution in much better way. But right now we rely on number average molecular weight by counting the chains.
Okay, okay, sir. So
Yeah.
my second question is like, you talked about this one-dimensional multi-component system where you are growing one component in elongation fashion, then another component on top of that. So if we co-mix these two components together and then grow it, then it will be like one-dimensional or it will be multi-branch like system.
Yeah. So again, nice point. There are, just like classical polymers, there are two ways we can do it. One is sequential, but if you have control on the reactivity ratios, we can do it in one pot as well. So we have demonstrated in the kind of secondary nucleation, we can do it in one pot at a time. No need to rely on the secondary nucleation, sequential addition. And in the first case also, we have examples where we did it, the organic heterostructures, one-dimensional organic heterostructure also. In one case, we did it in one pot. So it depends on the reactivity ratios of the monomers. So always safe option is do it sequentially in a kind of step by step. But then if we can control the reactivity ratios in such a way that we can also do it in one pot. So we have done that. Again, it depends on the systems. Yeah, I hope.
Okay, sir, understood. And
Yeah.
the last question is like, one of your this chirality induced growth. Like if I am correct, like the structure is somewhat twisted. This spiral-like structure was there. So is it really spiral? And if so, such kind of helical structure can we get?
Yeah. So Indeed, it's possible. So in some cases, definitely they translate into a kind of microscopic chirality. But then in all cases, it doesn't translate. So, so because they are chiral, we know that from circular dichroism, but that is at a kind of really at a primary molecular level. So in all cases, it's not necessary to be translated into a kind of microscopic chirality. But there are certain fibers, we do see that whatever at the molecular level and supramolecular level get transfers into a kind of even to a bundles or microscopic chirality. So this is not true for all the cases, but in some cases, we do see that.
Okay, sir. Thank you. Thank you so much.
Thanks, so yeah.
So already 8 minutes delay after 1 hour.
Sorry, I took 10 minutes extra for my talk. Yeah.
From our side also a lot of questions are there. Okay, so thank you, Professor, once again for taking our this what I will say, this wonderful discussion. And not hopefully, this is really that we have been benefited with some different kind of knowledge compared to our within our box. That is perfectly true. And some new ideas also what can be implemented in our cryo and related things. That is also one there. And thank you, Professor, and thank you all for joining. So we'll meet in the next month, and everything will be announced. We'll communicate with you all with a separate. Okay. So,
Thank you, Subrata. Let us also wish Professor Subi George all the very best to expand his science.
Thank you.
To much larger scale, and everything is becoming possible today. The dream that he talked about, expanding that further. All the very best. Thank you.
Thank you. Thank you so much. Thank you.